So another short little lecture here, mutations. So by definition, a mutation is any change in an organism's DNA. It could have some consequences, okay? It could lead to different phenotypes that could be bad for the organism, but it could also lead to phenotype changes or a different ability that could be beneficial. So mutations, some are bad, but they can be good. So one type of mutation is called a point mutation, which means a single site in the DNA, a base pair. You can either add or delete, or you can change the base from like an A to a T or C to a G or any other nucleotide. So base substitution is usually called a missense mutation. In other words, you're changing the amino acid of the protein. You could, anyways. So if you change the amino acid in a protein due to a base substitution, you call it a missense mutation. So there are different point mutations. So I just said missense, where you change it and you get a different amino acid. So that's missense. You can also have silent mutations because the code is redundant. If the original is AAG and all of a sudden it changes from AAA, it still codes for lysine, so it still codes for an amino acid. So yes, a mutation took place, but it coded for the same amino acid, so you get the same protein at the end. So we call it a silent mutation because there's no effect on the organism. It's just a change in the base pair that coded for the same amino acid. Nonsense mutations occur where instead of getting an amino acid, like lysine, it codes for a stop codon, which means that, depending on where this happens, it could be pretty harmful. Maybe it doesn't even, maybe it's like the second amino acid. Well, that would be really, really bad. You wouldn't even get a protein. So I think I go on to talk about all of them in detail here. So two classes of missense, transition and transversion. Basically, if you go from like pyrimidine to a purine or a purine to another purine, that's what transition and transversion is. So here for transition does not change the type of bases in the base pair. So a pyrimidine is substituted for a pyrimidine. So what are the pyrimidines in DNA? T and C. Yep. So if a T it turns into a C, or a C turns into a T, you have a transition. If you go from purine to purine, it's transition. So A to G, or G to A. Okay, so that's transition. Transversion means you go opposite. Pyrimidine substituted for a purine. So like you go from A to C. You get a transversion there. If the new codon from a base substitution encodes for the same amino acid, then that's where that silent mutation comes from. So here's another example of a silent mutation. CAA turns into CAT. It still codes for valine. So silent mutation, no. This diagram is showing the transition transversion. I know it's kind of blurry, but from A to C, transversion. From A to G, transition. Okay, case study with sickle cell anemia. Sickle cell anemia turns your red blood cells into sickle cell shape, and it's based on one base pair substitution. So instead of getting CTG, it turns into CAC, and you get valine instead of glutamate. So it's just one base pair mutation. The effects of this, oxygen isn't carried efficiently because it doesn't really have that ability to carry oxygen. A lot of times people are in pain or they suffer from anemia, blood anemia, low levels of red blood cells carrying oxygen. However, in Africa, so I know this diagram is blurry again or small, it's really, really popular in Africa versus other cases in the world. And the reason for that is to be a carrier of sickle cell anemia, yes, some of your red blood cells are sickled cell, but it actually has an advantage against malaria, which is another very prominent syndrome in Africa. Malaria works. It's basically a parasite carried in the saliva of mosquitoes. And And what this mosquito does is obviously gives the organism, the host, the parasite, and the parasite burrows into healthy red blood cells and like lay eggs and kind of chills out in there for a little bit. And then when the eggs or larvae are ready to hatch, it literally ruptures red blood cells. So then your red blood cells like explodes, you damage red blood cells, and the parasite escapes, and it affects more red blood cells. And so, like, if you lose red blood cells, then you suffer from anemia, you're in pain, so it's, it's got some harmful effects. Now, the parasite, if someone has sickle cell anemia, your red blood cells are sickled shape, and it kind of makes it rough and rigid. The parasite can't burrow into red blood cells. So, to be a carrier for sickle cell anemia, which means some of your red blood cells have that, is actually advantageous because you're less likely to suffer the effects of malaria. Makes them more resistant to malaria.
But then wouldn't sickle cell anemia cause some problems by itself?
Right, but if you're a carrier, which means you're heterozygous, the effects of it are not that great. Like, you could live with it. Yeah, you're gonna feel a little bit of pain, but you're not gonna be like on your deathbed.
Well, except for you might be given to your kids.
Right, and that's the thing about being a carrier. You do have the ability to pass it on to your child. Okay, so a nonsense mutation means the amino acid gets coded to a stop codon, and they call it nonsense because basically you just stop the process of making the protein. We talked a little bit about frameshift mutations when we were talking about, is DNA code a triplet, like the read-in frame, is it spaced or unspaced? But a frameshift mutation happens when you add or delete a single base. It alters the read-in frame, it shifts everything over one way or the other. If the mutation is early in the amino acid sequence, the majority of that protein will be altered. It can cause premature termination, which means that it could turn into a nonsense if it codes for a stop codon. Frameshift mutations can be caused by your environment. UV can induce frame shifts in your DNA, and we'll talk a little bit about DNA repetition, how DNA, when it replicates, it repeats regions, and so now you get like these triplet codes that just get repeated over and over and over. So there are actually some diseases that are the result of that. So this kind of, again, another summary of point mutations. This is the normal DNA. The missense, what's missense? Changes one.
Changes the amino acid.
Yep, changes the amino acid, thank you. And then we have our frameshift insertion where it'll shift the read and frame that way. Deletion, it will shift it this way. And then that nonsense is that stop codon. So let's focus in on that triplet repeat expansion mutations, which your book calls dynamic mutations. So in one case, your DNA might start to repeat a codon, in this case CAG, 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 CAG. And so in these disorders, these individuals, they have these triplet repeats in their genome, and they just keep like repeating themselves every time the DNA replicates. And then later on in life, if you have enough of those repeats, you've reached the threshold, if you will, then that disease starts to come about. And so one very popular triplet expansion mutation is Huntington's disease. Huntington's disease is a dominant allele, if you will, that causes these triplets in a person's genome. And people that suffer from Huntington's don't realize they have it until they're in their midlife, like late 30s, 40s. That's when they start to feel the effects of Huntington's.
Can people get tested for it or not?
Yes, you can get tested for it. Yep. So the sad thing is, if they've reached midlife and they realize they have Huntington's and they're married and they have children, they probably have passed on Huntington's disease to their children. Some other disease associated with triplet repeat expansions, muscular dystrophy, Fragile X syndrome, and Kennedy's disease. So I think I'm going to talk a little bit about the Fragile X syndrome and maybe muscular dystrophy. But
Which Kennedy was
Is-
that named
MS. Is that muscular dystrophy?
Yep.
So why isn't it called MD?
oh no wait, sorry.
after? MS
And
is multiple sclerosis.
sclerosis.
Okay.
Yeah, sorry, two different things.
My grandma had that, and my dad used to. Okay, she was in the nursing home, and my dad used to bring bees to like sting her,
Why?
to like I don't know, stimulate nerves or something. I don't know.
Oh. I'm gonna have to look into that. Fragile X syndrome. So it happens on your sex chromosome. Okay, the X chromosome. In this case, it's a repeat of CGG on a certain gene. In this case, the FMR1 gene. So because it's on the X chromosome, it's a sex-linked dominant matter. Some results from it. Males usually have abnormal faces, kind of a long face, large ears, and a very prominent jaw. Females, same, but just like milder features. And, out of 10,000 people, or males, I should say, 16 to 25, have Fragile X syndrome. It's all because of the CGG repeat. Huntington's disease affects motor, cognitive, and psychiatric changes. Usually happens mid-30s to 40s is when they start to feel some or experience some of these effects. The repeat, the triplet, is CAG on an HD gene on chromosome 4. And this is autosomal dominant. So this is not sex-linked. This is on chromosome 1 through 22, in this case chromosome 4. And so, yeah, you can pass it on. So this is a figure showing normal 5 to 30 repeats. It's okay to have 5 to 30. You're not gonna, feel or have Huntington's disease. But then, however, people that start to get those effects, they have 50 to 2,000 repeats in their genome. That's a lot. That's why some cases are way more severe than others. So as they live longer and longer, they get more triplet, repeats, and it will just get more and more severe. Switching gears to chromosomal mutations. So you can actually change the structure of your chromosomes. A lot of times it happens during meiosis, like how the homologous chromosomes pair up and then pull away from each other, or even mitosis just by being, again, pulled apart in wrong ways. But there could be errors in crossing over during prophase of meiosis 1, which could lead to gene duplications or deletions. So in our karyotype, some people had some deletions, or like one little bit was taken off and pasted onto another chromosome. So we'll talk about some of those diseases. Yeah.
They made it now like it's like create a baby, like where they gene edit the baby or like the
Using CRISPR.
embryo.
Yep.
That's so messed up.
Have you seen the movie Gattaca? Say we all do. Nope, that's why. Never mind. Because I think my original plan was to take the exam Thursday and then just like watch a movie. Gattaca is a great movie. It's a little slow, but it's about people that they're gonna have a kid. They go in and they're like, oh, what would you like your child to be? Well, I want him to be a really good pianist. So then they alterly change their DNA and maybe they're gonna give them 6 fingers on each hand so they can play more keys, or other things like that. Like you want them to be a professional basketball player. Okay, we're gonna go in and alter all the genes that affect height, so then that way you can.
We have a test for an exam Friday.
No, next one.
Okay.
Okay, so one type of chromosomal mutation is deletions, which means you're going to lose part of your chromosome. So one disease is cry de chat, which is French for cry of the cat. And in this case, we have a deletion on chromosome 5 at the very top, and it's just a little bit. But as a result, feeding problems, poor growth, severe cognitive and speech, as well as motor delays, behavioral problems, excessive drooling, small head, and jaw. And it's all because of a deleted part on chromosome 5. Duplications may or may not lead to phenotypic consequences. So again, it happens in meiosis when the homologous chromosomes misalign with each other, or during DNA replication, where we have replication slippage. So what happens is we have DNA polymerase, which helps replicate DNA, and all of a sudden it replicates this, and then all of a sudden it falls off for some reason. So then it goes back and restarts, and it reads the same 3, and it adds those 3, and then it falls off again. And now we just keep adding repeated segments into our DNA. So they call them tandem repeats. And so as a result, you can get duplication of genes that could be bad, or it may not have no effect at all, depending on where that's at. We'll talk about, like our genome later on in this unit, but you only use like 5% of your DNA. And so like the rest they call junk DNA, and they don't know really the purpose of junk DNA yet. My husband has an interesting theory about it. He's kind of out there with his idea.
What is it?
He thinks that we're huge fans of Ancient Aliens.
Alright.
Yeah. And he thinks that junk DNA is like alien DNA. Yeah. So you'd have to learn more. You'd have to watch Ancient Aliens to kind of get the full premise of it. Could be.
Potentially.
I think it's legit. But, Okay, inversion happens when there's a break in the chromosome, and it literally just flips around. So instead of being A, B, C, D, E, F, G, H, I, now you're A, B, C, F, E, D, G, H, I. So again, it may have an effect on the phenotype, or it may not. But it does happen. The last chromosomal mutation that could occur is translocation. And so this was on our karyotype assignment for some of you, where one part of a chromosome breaks off and literally switches part with another chromosome. So in this case, maybe chromosome 4, this part's going to break off. Chromosome 20, this part's going to break off. And so they like crossed over when they should not have crossed over. And so now this part of chromosome 20 is on chromosome 4, and this part of chromosome 4 is now on chromosome 20. So it's all about meiosis and how they correctly match up with each other. In this case, they did not match up correctly. So you're moving genes from one chromosome to another. This is actually how leukemia comes about. And it's just because genes on different chromosomes, they translocated, and now they are expressed inappropriately in blood cells and lead to cancer. All right, now I said a lot of times mutations, they can be bad, but they can be beneficial. And so mutations, yes, you get a change in the DNA, which could lead to a change in the phenotype. But as a result, you are producing new alleles and therefore new traits. At the population level, which is a group of species, of the same species living in an area, it does make evolution possible. It does make evolution possible. Because we have different variations, sometimes people will be more, have an advantage over their peers. And so they will survive and reproduce and pass on. So they have a higher fitness because of that mutation, if you will. At the individual level, if the mutation is bad and it, causes a disease or death, obviously that's going to lead to a decrease in fitness. Okay? But it could, like I said, be beneficial and lead to an increase where it will be passed on to the next generation. And so you get evolution occurring. Now the last 2 slides for the lecture, I just made a table of all the mutations. So this is point mutation. We talked about silence, where it codes for the same amino acid, tryptophan, tyrosine. Missense means you get a different amino acid, changes the primary structure of the protein. Nonsense means you get the stop codon, and then a frame shift is insert or deletion in the genome. We also talked about gene duplication, which means that things just get duplicated over and over and over, so you can get Huntington's disease. And then chromosomal inversions, where it breaks and it flips. So it's just a nice little summary of the mutations we've talked about. So at the end here, we have some mutation questions. We have some multiple choice. Now this right here, question 4, is probably going to be the longest for you to answer. I give you a DNA sequence. This is the original, and this is the strand. You have to put it to mRNA because there's no T in mRNA, so you have to write out the mRNA
GC.
No,
GC
Oh yeah.
A, yeah.
A.
And then from there, code for the amino acid to figure out what the mutation is. So it's kind of tedious. In your book, there's a table of the amino acid codons, or you can Google it. That works as well. So you have to convert it to mRNA, then figure out the amino acid sequence, and then, what kind of mutation is it? So basically, how does it affect the
So that's like 3 questions right there. Like
Yep,
those
A,
are all
B, and C.
3
Yep, they're different. So
from
you can
the-
tell, like, this one for sure has an insertion, because so that's the mutation, but then you have to show how the mutation affects
Does
it.
C have an insertion too?
Possibly. It could be a repeat. I don't know. I'd have to look at my notes. I mean, I have my key, but it's just... 5 is multiple choice. 6, what would happen if a mutation changed a start codon to some other codon? And then, so there's only 6 questions for your assignment, but then I had to share, so if the normal word is beast, and all of a sudden you change the B to an F, you get feast. If you insert a letter, you could get the word breast. You could delete an A and get best, and then inversion